Hi there, today we're going to look at immunity or immunology, including T cells, B cells, and basically the specific immune response. So we're going to start off with the first step, which is usually there's been some type of introduction of bacteria or a pathogen, I should say, to an organism, and it's managed to get past the first barriers. So these pathogens are going to have an antigen sort on its surface, and in this case I'm going to draw my pathogen with a very large red triangle antigen. Obviously this is not to scale. What's going to happen first is you're going to have a non-specific immune response, which is engulfing via a macrophage, which is known as phagocytosis. So a macrophage is a large white blood cell which changes its membrane shape so that it can engulf the bacterium or pathogen. So this is a macrophage, also known as phagocytosis, a phagocyte, sorry, and this process is phagocytosis. It's engulfing the pathogen. It doesn't know exactly what kind of pathogen it is. It just knows that it doesn't belong in the organism. That's why it's known as non-specific. Okay, what happens next? Once it's inside the pathogen, it forms a vacuole, and the pathogen will actually be digested, so that will be broken up into bits, but the antigen itself is kept intact. And the antigen is going to be presented on the macrophage cell membrane surface so that it can be presented to other white blood cells. How does it do this? Well, it has a special apparatus known as a major histocompatibility complex. And this is kind of an apparatus which sticks out from the cell surface membrane and presents the antigen to the rest of the immune cells. Let's just draw that like that. There's the antigen, like so. And we've got the apparatus that presents the antigen called the MHC, or major histocompatibility complex. That is a long phrase. Okay, so what is this called now that is presented this antigen? Now that it's presented the antigen, it then becomes known as an antigen presenting cell. Or APC for short. You can put either in the exam, the abbreviations or the full term. Okay, what comes next? So we're presenting this antigen to the rest of the specific immune cells, and so we need to activate those other cells. So along comes our first other white blood cell, a type of T cell known as a T helper cell. And these will just be present in the blood or in the organism's body. And there are several of them there that will have different shaped receptors on their cell surface, and one of which will match and be complementary to the actual antigen that we have here. So the receptor on the T helper cell is complementary in shape to the antigen that is now presented on the antigen presenting cell. So once it's bound to that, the T helper cell starts to release a chemical known as cytokines. I'm going to draw these in pink. So cytokines begin to release, and I'm going to draw some this way and draw some going up this way because I'm going to show two routes now, the cell-mediated response and the humoral response on the same page. So these are cytokines, and these cytokines are going to activate B and T cells, or T killer cells rather. Let's deal with the T killer cells first of all, and we're going to go up all the way over here, so let's show the cytokines moving up in that direction. Okay, so the cytokines are going to activate some T cells. Let's go with our T cells also being green. So in the body, in the blood somewhere, you will have a number of different T killer cells waiting to be used. So what we have essentially are different T killer cells. T killer, and T killer, all with slightly different shaped receptors on them. Like so. Now, out of these ones, the cytokines are obviously going to be activating the T killer cell that has the correct receptor, which will be complementary to the antigen on the antigen presenting cell. So out of these four that I've just drawn, which one is the correct T killer cell that's going to be selected? Correct. It's going to be the second one, because that's the most, according to my drawing, complementary to our antigen, a sort of triangle shape. So this one is chosen, and that has a special name, that process, called clonal selection. Okay, we have the correct receptor for our antigen. What's going to happen next is we only have one of these, so we need to make more of them. So how do we make more of them? They basically clone themselves through mitosis. And here we're going to make more of these T killers. We'll also make some T memory cells here as well. So I'm going to draw a couple of those over here. So let's just draw on the actual receptor to show that they've been cloned. And it's really important that the genetic material is identical, because otherwise that particular receptor, which is likely to be a protein, won't be produced correctly because the gene may not be correct. So it has to be the correct gene, and therefore identical to the one previously. So these are T killers, and these are T memory. Now, what happens with the T memory cells? These are going to stay in the blood. And those are there ready to be activated again, should someone be reinfected with the same pathogen, with the same antigen. So they're there ready. And so the T memory cells are providing long-term immunity. Okay. Right. So what happens though with the current pathogens that are in the system? The T killer cells that have now been made are now going to do direct killing. So what these will do is they will go and find pathogen that has the correct receptor, and it's going to bind to the pathogen, like so, with the antigen. As you can see, my antigen is getting bigger and bigger. And obviously, these are not drawn to scale. But it gives you a clear idea of what should be happening. The T killer cell will then bind to the antigen on the pathogen, and it will release chemicals which will cause damage to the cell wall of our pathogen if it is a bacteria, and it will damage the cell wall and so cause water to move into the cell. And by osmosis will eventually fill it up and cause it to burst. So these chemicals damage the cell wall. Of bacteria, I should say. Viruses would be a different, slightly different mechanism. Okay, so this is a direct kill or destruction, I should say. Okay, so that's T killer cells. T memory cells, like we said, they stay in the blood and they'll provide long-term immunity for the next time. Right, let's have a look what happens with the B cells. So with the B cells, we're going to use a different color here. So I'm going to use purple. Same thing, cytokines are released, and it all starts off with the same process. Cytokines released, and again, we have a kind of well or reserve of B cells, which don't technically have a receptor, but I'm going to use it here, give you the idea. But they have the correct genes to make the correct antibodies that will be complementary to that antigen. So just for the diagram's sake, I'm going to add receptors here, but they're not really present on B cells. Okay, B cell, B cells, B cell, and B cell. Okay, so according to the ones I've drawn here, which is the correct one, that's going to be activated to bind to the antigen. Correct, number 3. Again, this is called clonal selection. And then the next step is, of course, that we're going to have cloning and more memory cells as well. So we're going to get some more B cells. And I'm going to change that to a different color so we can see the memory cells. And they're going to clone and make more of themselves. So I forgot to mention over here, actually that cloning is by mitosis. And that process is known as clonal expansion. And it's the same here, mitosis and clonal expansion. Okay. And we've got our memory. B memory cells. And more B cells. So what happens to the B memory cells? You could probably guess. They're going to stay in the blood. Like so. And the actual B cells themselves are going to do something different. We don't have a direct destruction of the pathogen here. They're actually going to now change into a different type of cell. And that B cell is going to, what we call, differentiate into a different cell called a plasma cell. So, now what do the plasma cells do? These produce, or actually produce, the antibodies. And they produce millions of them. Now, the antibodies are a Y-shaped protein, and you might have seen this somewhere before. They have 2 antigen binding sites attached to common chains here. They're made up of 4 polypeptide chains altogether, but 2 antigen binding sites. The antigen binding sites will be complementary to the antigen. So they make millions of these Y-shaped proteins. And the only way we can get proteins is if we have the correct genes that are in the actual DNA itself. So that's why everything has to be identical. So we can get the correct gene, the identical gene that is cloned from plasma cell to plasma cell, of course, and therefore we get the right antibody being transcribed and translated. So if we look at just quickly how the antibodies work, I'm just going to draw this more simply over here. They kind of work through sometimes clumping pathogens together, also known as agglutination. So what we would have is a pathogen could bind to that one, could bind to that one, could bind to that one there, because it's two antigen binding sites for each. And you can imagine we have the little triangle on each of those, and so on, like so. So this is called clumping or agglutination. This is just one method by how antibodies can help to get rid of the pathogen. And the antibodies don't themselves actually destroy the pathogen. What they're doing is they're clumping them together through agglutination so that a phagocyte, such as a neutrophil, can come along and engulf these much more easily, including with the antibody itself. So basically, it's making it easier for phagocytosis to happen. Other ways that work as well are through the neutralizing the antigen. Sometimes the antibodies are used simply to just cover up the antigen so that the antigen can't bind to a host cell in the case of a virus, so it can't unlock the access to the host cell and therefore inject its own genetic material. So just to finish off, to name a few things. So first of all, anything involving the T killers is called the cell-mediated response. Anything involving the B cells is called the humoral response. Okay. And just to add on extra little bits here and there, throw a spanner in the works, just be aware that the T helper cell is a kind of go-between the humoral and the cell-mediated response. It's kind of a middleman. So it can activate both the cell-mediated and the humoral response. On top of that, another spanner, B cells are able to bind directly to the antigen presenting cell antigen. They don't always need the T helper cell to activate B cells. They can do it directly. And there we go. That's our cell-mediated and humoral response with T cells and B cells and phagocytosis. Hope that helps.